I will now present the project of Peking iGEM 2017. This is a video of zebrafish embryonic development. Cell division is controlled precisely in time order. The case is Drosophila development. The beautiful pattern of cell movement also varies over time. As we can see from above, cells perform a series of functions in sequential order. This is the fundamental phenomena in biological world, the life process. Just imagine when we can design life process, what can we do? We can achieve periodic drug delivery so that drug release can be done precisely in sequential order at appropriate times. Design automated cell factories that divide release products and lysis automatically in pre-programmed procedure, all in one fermentation tank. Cases above, the cells complete multiple missions in specific order. In the viewpoint of digital circuit theory, this is called logic. Sequential logic circuits would enable new approaches to the study and control of biological processes. In other words, we can unfold a new dimension in designing synthetic life. This year, the Peking iGEM team attempts to develop a framework of programmable biological sequential circuits. In electronic engineering, sequential logic is a mature concept enabling very complex computations. An sequential system is made up of several basic components. No matter how complex it is, an electronic sequential system is made up of several basic components. Using design procedures, they can be assembled to complicated systems such as cell phones and In order to know how it works, let's see a simple example of digital album. The pictures will periodically and automatically display in order. What is the mechanism? There is a periodic trigger signal serving as fundamental clock, but it has only two alternating states, high and low. If we add a frequency divider to generate a new one with a period twice as long, then consider the high and low status of two signals together, we have four states. An interpreter is used to translate the signal states into functions, which in this case, the sequential display of pictures. To sum up, there are mainly three parts for sequential logic. First, clock, an oscillator that generates a periodic trigger signal. Frequency divider, the most important device that stores state information. They are so important that every digital system requires billions of them. The third part is the interpreter that reads the state's information of frequency divider and change it into functions. If we apply all of them to biological system and then follow standard steps as what we do in electronics to design, we will reach the aim of sequential logic. Now we will introduce its counterparts we designed in the biological system. First, we must have a frequency divider in the cell to generate different state information at different times. There are two requirements. The frequency divider is designed to respond and change its state only when the original signal comes up. Otherwise, there is no change in state. How we can implement such device in living cells? The answer is DNA. People have used DNA to store information and encode functions in DNA sequence by flipping DNA fragments. Recombinases flip the sequences between ATTB and ATTP sites. Then the sites will change into ATTL and ATTR. When recombination directionality factor RDF is also present, it can flip back the sequence. We consider genetic divider with two recombinases and recombinase RDF fusion proteins on two strands. The proteins on one strand control the promoter direction on the other strand. Recording the status of promoter directions as states, and we can realize the function of electronical frequency divider. First, we induce the upper promoter, a recombinase is expressed, and the lower promoter is flipped. We get our. Then induce the lower promoter, a recombinase for the upper sequence is expressed, the upper promoter is flipped. We reach the third stage. Both promoters are now in reverse direction. Next, we induce the upper promoter again, and recombinase RDF fusion protein for the lower sequence is produced. This will flip back the lower sequence. This is the fourth state. Finally, the lower promoter and the upper one will be flipped back to its original direction. We come back to the initial state. To summarize, the frequency divider has 4 states in total. We pack the trigger signals into 2 groups, each consisting of 2 signals for the different promoters. The period of its state is twice the same length as that of the trigger signals. Thus, frequency dividing is achieved. Use the interpreter to translate state into function. In our design, the interpreter is a sequence with recombination sites on it. Between the sites are functional sequences such as terminators, promoters, and coding sequences. Flipping them will alter the transcription direction and thus change. Finally, we would like to trigger the state transition automatically. We need 2 alternating trigger signals. The repressor implements long-term stable oscillation with 3 mutually inhibiting repressors. The level of repressor expression and the corresponding promoter strengths are oscillatory. It has 3 signals, and our system takes 2 of them. In all, we use the repressor as a genetic clock that precisely generates gene expression. Pulses. The pulses change the state in recombinase-based gene frequency divider. Each state of frequency divider can be read out by the downstream interpreter driving cell function. With these designs, we are now ready to design live processes. Then, we'll show you more details about experiment frequency divider. To build the frequency divider in biological system, we choose 3 recombinases from phages 5C31, BXB1, and TP901 because they have high efficiency in gene recombination and have wide applications, and most importantly, they all have well-studied RDF proteins. This is our experiment setting of recombinase characterization. Because the function of our device involves flipping promoters, we designed a constitutive promoter flanked by recombination sites ATTB and ATTP. The promoter is before recombinase expression is induced, the cell expresses GFP. After induction, the promoter is flipped, recombination sites changed into ATTL and ATTR, and cell fluorescence shifts to red. We observe fluorescence shift through flow cytometry. On the result diagram, X and Y-axis represent red and green fluorescence intensity. So we dissect the plane into 2 regions. Cells with flipped promoters should locate on the lower right region, and those with promoters not flipped locate on the upper left. However, when we did preliminary tests, we found that the flipping is insufficient. Most cells contain promoters that are still in the GFP direction. Therefore, we must do fine-tuning. There are 2 possible levels of adjustments. On transcriptional level, we use promoters pBAT and pTAC. On translational level, we generated an RBS library. Part of them are from iGEM parts registry, and others are first analyzed in RBS calculator and then synthesized de novo. Here are flipping efficiency results of TP91. We select an RBS with high efficiency. Then we did the same to BXB1, and flow cytometry results show sufficient flipping. C31 has rather high flipping efficiency. Still, we need to test and find a promoter RBS pair that has low leakage. We also did characterization of recombinases and plotted flipping efficiency over inducer concentration. This will provide data for parameter estimation in modeling, and also suggests the inducer concentration in further experiments. We then did similar characterization to recombinase RDF fusion proteins. Fluorescence shift is set opposite as before, from RFP to GFP. Theoretically, when no RDF is present, the recombinase itself cannot flip back the sequence fragment, but we observed a significant portion of the cell group expressed GFP. We conclude that recombinase RDF fusion proteins are functional, but needs more fine-tuning work. With characterized recombinase and fusion protein parts, we are ready to assemble them and construct the genetic frequency divider according to our design. We first did simulation of the whole process to prove its viability. The four flipping reactions are modeled in detail by ordinary differential equations, and sensitive parameters are fitted with experiment results described above. Here's the simulation result of the whole process of frequency dividing. On the slide shows changing of promoter directions over time. While adding 4 successive inductions, the model system reached the 4 states as described in our design, so proof of concept is successful. We then verified the 2 flipping reactions in experiment. GFP is co-expressed with recombinase, so when we add the first inducer arabinose, recombinase BXB is produced and the cell shows green fluorescence. The lower promoter P tag is flipped. When we add the second promoter inducer IPTG, recombinase TP901 is produced. The first promoter is flipped, and thus GFP transcription is closed. After the first induction arabinose, GFP is expressed, and after second induction IPTG, green fluorescence is off. We then updated the simulation model to incorporate repressilator. We substituted the inducible promoters PBAT and PTAC with inhibitory promoters PTAT-O and PR. These promoters will generate protein expression pulses as they do in the repressilator. The simulation result proves that, firstly, the inhibitory promoters used in the frequency divider can be driven by the repressilator. The promoter strengths are high enough for flipping reactions. Secondly, the system also reaches the 4 states, as in the case of inducible promoters. The third part of our system is the interpreter. Cell states are specified by the frequency divider, and it's the interpreter's task to convert state into function. As the cell transits from one state to another, different functions can be executed. The task of the interpreter is to incorporate functional DNA sequences into recombination sites. On the different states, the expression of recombinase or recombinase RDF fusion protein varies. Thus, the directions of these sequences are changed during each state transition, and cell function is altered too. Take an example. Terminators can block RNA polymerases, blocking transcription. By flipping terminators by recombinases, we can theoretically switch on or off the downstream gene. First, we need a series of unidirectional terminators. As shown on the slide, when a terminator is inserted into the sequence in forward direction, it will block RNA polymerase and turn off GFP expression. When it is inserted in reverse direction, it will allow RNA polymerase to pass and turn on GFP. So, how can we evaluate and select unidirectional terminators? We constructed 3 structures. Terminator is inserted in forward or reverse direction. In the third one, no terminator is inserted. Terminator strength, TS, is calculated by fluorescence intensity ratio of the sequence with no terminator over that with terminator. So we hope to select unidirectional terminators that meet the following requirements. TS forward is relatively high, and second TS reverse is approximately 1. Furthermore, forward TS exceeds the reverse one by at least an order of magnitude. tested and calculated forward and reverse TS to our library of 24 terminators and got the 5 ones that meet the requirements. The 5 selected terminators and calculated their TS ratio of forward over reverse. One terminator, 435 for short, has a ratio of over 1000-fold, which can be considered a well-performed unidirectional terminator. To verify unidirectional terminator can be inverted to change the gene expression, we constructed 2 reporter plasmids. As for type A in initial state, the terminator is in reverse direction and it can express GFP. After flipped by recombinase, the terminator will block transcription, then turning off GFP. Type B cell behaves oppositely. The terminator is flipped from forward to reverse direction. Green fluorescence is first off then on. Apart from these 2 types of cells, we add another type of cell that constitutively expresses GFP. Then we deploy these cells onto a 96-well plate as shown on the slide. Before the recombinase flips the target sequences, green fluorescence shows the pattern of 2017. Recombinase-mediated flipping, fluorescence pattern changed into PKU, short for Peking University. In experiment, we induced the expression of previously characterized BXB1 recombinase. After 12 hours of induction, fluorescence display shifts from 2017 to PKU obviously. This indicates the terminator was flipped efficiently by BXB1 recombinase. In our project, we hope to use recombinase expressed in every state to control the cell's function. This requires cells can realize state transition from more than one. So how can we design this complex interpreter circuit? As a proof of concept, we designed a fancy fluorescence display in 96-well plate. We used 4 types of cells to show the fluorescence pattern from 1 to 2, then to 3. Besides the cell to constitute GFP, we need 3 types of cells to show its transition. The cell A shows an GFP pattern that from 0 to 1, then to 1, and fluorescence of cell B 0 to 1, then to 0 again. C shows the pattern from 1 to 0 to 1 finally. We found that there are so many possible circuit designs for every of these 3 types of cells. It's difficult to design them manually and select the best one. So how to design interpreter circuits for more complex sequential logic? We designed a software called Carpiot to design, evaluate, and select the relatively better circuit. Users only need to define the state numbers and the cell functions in every state. Our software will generate the selected circuit based on a lot of rules and data, such as the performance and characterization of recombinase, the properties of some functional DNA units. Through Carpiot, we get all possible for our designed fluorescence display and the best one circuit for these types of cell. Our characterized TP901 and BXB1 recombinase to flip the target sequence successively. We can see fluorescence transition from 1 to 2. From the experiments and demonstrations above, we conclude that our framework is capable of implementing sequential logic in living cell, and we strongly believe that with further development, more complex functions of cellular machines are within reach. When we were pushing forward our project, we met several problems on information collection. For example, where can we get detailed information about the recombinases and RDF proteins? Authoritative sources like PubMed and all official websites, Wikipedia, all have their drawbacks because they are not targeted directly to synthetic biologists and iGEMers. Therefore, the Peking iGEM team built a powerful and precise knowledge repository that is dedicated solely to synthetic biology, that is symbiowiki.com. From normal wiki functions such as wide coverage and editing, wiki is born with strong social capabilities. You can not only acquire and contribute information, but also make friends and discuss with synthetic biology researchers and iGEMers worldwide. Together we are a community, and we would like you to join and make this happen. Don't hesitate to visit the website right after this presentation. This year, our project is highly interdisciplinary. The future of combining electronics, technology, and synthetic biology is brilliant. We interviewed professors of synthetic biology and electronics engineering on theoretical basis of our project. We held several seminars and educated students on different levels about overlaps of the two fields. We also wrote a brochure to provide a brief introduction to electronics for synthetic biology. This year, we co-organized CCIC, a special event for iGEM teams in China. We also interacted with several undergraduate and high school teams for academic issues, project discussion, laboratory techniques. Our teammates for their industrious work 6 months, and we give special thanks to our instructors and those researchers that offered us precious help and guidance. Without them, this project would never have come this far.